Assalamualaikum. Myself, Maryam Batool, and I am here from Department of PS Biochemistry. Today, I am going to represent the topic of my presentation, glycyrrhetinic acid. Let's look have on the contents, please. Introduction to glycyrrhetinic acid, methods of preparation, physical properties, chemical properties, applications, summary, and references related to the glycyrrhetinic acid compound. Introduction. Glycyrrhetinic acid. Glycyrrhetinic acid is basically a hydrolytic product of glyceric acid, is a component of licorice, and causes apparent mineralocorticoid excess by inhibition of the enzymes 11-beta hydroxysteroid dehydrogenase, which mainly converts cortisol to the inactive cortisone. Mineralocorticoids. Mineralocorticoids are a class of corticosteroids, which in turn are a class which in turn are a class of steroid hormones. Mineralocorticoids are produced in the adrenal cortex and influence salt and water balances, electrolytes between balanced fluid in our body. The primary mineralocorticoid is aldosterone. This enzyme activity is present in the kidney. Inhibition induces local cortisol excess, which binds to kidney mineralocorticoid receptor and exerts a mineralocorticoid-like effect for 19. As a result of blockade of cortisol mechanism in kidney, urinary cortisol is increased, plasma cortisol is unchanged. Thus, the urinary cortisol is falsely elevated in subjects taking excess quantities of licorice. Preparation of glycyrrhetic acid. Glycyrrhetinic acid, one kind of atropine saponin, is mainly extracted from the roots of traditional Chinese medicine glyceria. Students have shown that GA has obvious antiviral and anti-cancer effects and is commonly used for clinical treatments of chronic hepatitis and liver cancer. GA liposomes were commonly prepared by using lyophilization monophase solution method. Lyophilization is basically the method of forming glycyrrhetinic acid. Lyophilization or freezing dyeing is a well-known recognized method used for obtaining stable biological drug products that have short shelf lives in the solution. It is a low temperature process. Thermal degradation is minimized. Monophase solution basically a type of matter or a compound that contains only one phase or a clear-cut and unattached type of matter. The other method used for the preparation of glycyrrhetinic acid are GA also produced through metabolic engineering Saccharomyces cerevisiae by introducing the entire heterogeneous biosynthetic pathway of GA. Through this process, the codons optimized are combined with the beta-AS amyrine synthase and NADH, DPH, cytochrome P450 reductase gene of cerevisiae were introduced into Saccharomyces cerevisiae. The resulting citrine produces a chain of beta-amyrine and glycosyrrhetinic acid. Here we can see in the figure FPP sequence combines with the BAS through which form beta-amyrine 1, which reacts with the further and produces glycyrrhetinic acid 7A, and when it reacts with the UGATS, it produces glycerine 8. Single oxidation reaction catalyzed by CYP-GT8S single oxidation reaction catalyzed by CYP72A154. The cytochrome B5 from G urinalysis was identified, and this increased the efficiency of production of GA. Saccharomyces cerevisiae were overexpressed in a stable chromosome integration to achieve higher glycyrrhetinic acid production. Now, we should continue towards the physical properties of the glycyrrhetinic acid. Glycyrrhetinic acid is a hydrolytic product formed from glycosylate acid. The chemical formula of glycyrrhetinic acid is C30H46O4. The molecular mass given of the glycyrrhetinic acid is 470.7 gram per mole. Melting point is 296 degrees Celsius. The boiling point of glycyrrhetinic acid is very high and is given as 588.3 degrees Celsius. Solubility. It is, as we discussed earlier, it is a hydrolytic compound. It is soluble in organic solvents such as ethanol and dimethyl formaldehyde, which should be purged with an inert gas. It is sparingly soluble in acidic buffers. Abundance of glycyrrhetinic acid. There are abundant GA receptors on the cellular membrane of hepatocytes and hepatocellular carcinoma HCC cells. The receptor must perform a binding function. As we all know already about receptors, receptor performs a major function in our body. Receptors present on the skins of our eyes, receptors also present in our kidney, lungs, and other major organs in our body. Here, the abundance of glycyrrhetinic acid must present on the receptors where it performs a binding function. The histone-like DNA protein is one of the most abundant nucleides associated compounds industrially selected for the gene expression. Gene expression must be done by the process of DNA replication and DNA translocation. DNA replication and DNA translation processes must be done by the binding of proteins. Binding of proteins is mainly associated with the gene expression that is finally controlled by the glycyrrhetinic acid. Further, GA inhibits also methicillin resistant Staphylococcus survival and attenuates virulence gene expression. GA inhibits methicillin-resistant Staphylococcus. For example, GA also controls and inhibits the process of bacteria Staphylococcus and virulence gene expression. Other product name. GA is also known as urolinic acid, biosone, or enoxolone. As we see, the formula of, as we know, the chemical formula of glycyrrhetinic acid, here the structure is shown in the figure. The chemical properties include hydrogen bond donor and hydrogen bond acceptor. The number of hydrogen bond donor in the structure are 2, whereas the number of hydrogen bond acceptor are the number are 4. Exact mass. The exact mass of the glycyrrhetinic acid I molecular mass or exact mass molecular weight expressed in gram per mole is 470.73. Complexity. Here we can see the structure of the glycyrrhetinic acid. Many carbon atoms and hydrogen, oxygen are correlated with each other. The complexity found between the atoms of the glycyrrhetinic product is relatively high and is 96.5. Density. As GA found in hepatosomatic cells and cellular membranes, the density is 1 multiplied 10 raised to the power 5 cells per wall. As GA found in the cell membrane and hepatocytes and cellular membranes, the density and the thickness of the cells is 1 into 10 raised to the power 5 per cell wall. Optical density. The optical density of glycyrrhetinic acid 10 into 10 to the minus 6 and is very low. Glycyrrhetinic acid is a hydrolytic compound. When it is hydrolyzed, it is many structural derivatives are formed by the glycyrrhetinic acid. Glycyrrhetinic acid 4 to 9 derivatives are shown in the figure. Glycyrrhetinic structure derivatives of Ga 10 to 15 are shown in either COCH3, where only methyl groups are changes. The number of carbon atoms and methyl groups are increases, due to which derivative number are also increasing. Application Ga used in various fields nowadays, and the basic functions of the glycyrrhetinic acid is used nowadays is very common. The major application of glycyrrhetinic acid in liver cancer targeting therapy. Liver cancer. As we know, liver cancer is one of the most common cause of cancer mortality worldwide. It is most dangerous for human health and other. In this perspective, Ga, a pentacyclic triterpenoid present in roots and rhizomes of licorice, has been used as a ligand for targeting the liver cancer due to expression of Ga receptors on the suicidal surface of mammalian hepatocytes. In the perspective liver cancer treatment, Ga was used because due to its receptor cells, which works as the ligand for the mammalian hepatocytes present on the cellular membranes, which works as a target for the liver cancer. C11 carbonyl and C3 hydroxyl groups of the Ga have limited influence on the targeting action of Ga to HCC cells, and beta configuration hydrogen atom C18 contributes most targeting. As we see in the figure, on the presence of hepatocellular membranes, we can all see the presence of Ga acids, endothelial cells, which inhibits the growth of cancer cells, immunopotential of cancer in microenvironment. For example, free combined drugs are produced, activated drug delivery of drugs are produced on the surface of the cells, which reduce the liver cancer chances today. Other application used is the formation of glycyrrhetinic acid-based hybrid molecules. 5-fluorouracil is a cell cycle specific that interferes with the DNA synthesis and inhibits RNA synthesis. 5-fluorouracil attached with the glycyrrhetinic acid to provide mono or diconjugates of the pentacyclic terpenes with Fu resulting Ga5Fu and Ga5FUG. Alkyl chains 6 to 8 methylene groups. New hybrids have improved anti-tumor activity in case against cancer cell lines. This is shown in the figure A. GAFU6 are 8 methylene groups, and in figure B, we can see they are attached Ga5FUGA. In figure C, the new hybrid formed furoxane Ga glycyrrhetinic acid. Hybrid molecules have improved anti-tumor activity against cancer cell lines. Cancer cell lines are basically present. Other application of the glycyrrhetinic acid, which is highly selective dye removal and used nowadays very commonly, as we have seen in today. GA highly selective dyes removal. Supramolecular assemblies performed by the GA09 glaters in an orderly rod structures by stacking of pi by pi pyridine groups and the hydrophobic interactions of GA skeleton. Supramolecular gel attached with the pi by pi stacking through copper ions. Copper ions serve as a bridge in the adjacent rod structure. Formation of network of rod-like structure leading to gelling, solvents being extended to the gel volume declining, shrinking, absorbed. As we have seen in the figure, supramolecular structure are made up of nanofibers of pi by pi stacking of GA09 chelators. The copper ions present in the pi by pi stacking produces rod-like structures, which produces cross-linked nanofibers, and hence the shrunken gel produced and shrinkage observed. OGA used in covered viruses. The other major application of glycyrrhetinic acid used nowadays, which is very common and which is very valuable, successful for the technology and today evaluation, is covered virus. Glycyrrhetinic acid molecules are composed of a hydrophilic part, two glucuronic acid molecules, and a hydrophobic part. Glycyrrhetinic acid, when subjected to hydrolysis, releases 18 alpha and 18 beta glycyrrhetinic acid, which are the structural derivatives of glycyrrhetinic acid. During anti-inflammatory action mechanism of GNA, such as inhibition or translocation of nuclear factor NFKB and suppression of TNF-alpha, several in vivo and in vitro methods describe the use of GA in protection and treatment. Due to this, it is used for the treatment of severe acute respiratory syndrome, SARS-CoV virus 2. Structural derivatives of glycyrrhetinic acid, 18 alpha and 18 beta glycyrrhetinic acid, promotes the process of inhibition or translocation of nuclear factor and suppression of ENF alpha, which is used for the treatment of COVID virus. Summary. G is a hydrolytic compound wave from GL, which is basically licorice, a compound which is sweeter than the sugar and used in many other fields. G demonstrated as excellent cytotoxic, which works as a toxic material, antimicrobial, enzyme inhibitory, anti-inflammatory, antioxidant, analgesic, and antiviral effects. GA works as a selectively dye removal. Therefore, GA have become an active pharmaceutical ingredients. The references linked with the information was written in the presentation are given below. Thank you so much for your attention. Please keep good and responsible. Thank you.